Hi everyone, I hope you're doing well. Today we're going to be talking about Immune Technologies Lesson 11. So we're going to start off today by first checking our answers to yesterday's antibiotics worksheet that is attached below in the assignments. We're then going to be filling out our notes for the lesson using the teacher PowerPoint slide that's also attached, the videos that are embedded right within the PowerPoint slide, and the video that you're listening to right now. And then, after you're all done with that, we're going to complete uses of monoclonal antibodies handout that is also attached below. You're going to be using some links that are within this Google document and the information that we're going to be learning about today to answer some of the questions that are in this document. So let's get right on into it. I'm going to ask you to open up your notes to the page that looks like this. That is going to be the start of lesson 11. So for lesson 11, we're going to start off with monoclonal antibodies. And I'm going to ask you at the end of our discussion with monoclonal antibodies here, I'm going to ask you to go to this YouTube video to get a nice overview of everything that you should know about monoclonal antibodies. Therefore, let's go to our first PowerPoint slide for monoclonal antibodies. So you may have heard the term used before. You may have heard it in discussion or in conversation. You may have also heard it while listening to the news. It's a very interesting and popular topic in the science world and in research. So let's get into it. What exactly are monoclonal antibodies? So when an immune response occurs, antibodies specific to the pathogen are produced. And we already know this. We've already talked about how the body produces antibodies specific to antigens that could be on the surface of a pathogen. And we can make use of this in medicine and biotechnology by producing large numbers of antibodies in the laboratory to be used therapeutically or diagnostically. So, whereas an immune response may be polyclonal, so sometimes B cells could respond to multiple epitopes or multiple antigens, we also need monoclonal antibodies, in which the antibodies will be specific, very specific, to a specific pathogen. And here are some examples of monoclonal antibodies that have been used in treatment and to help with diagnosing different diseases. So therapeutically, an example would be emergency treatment of rabies, and to help with the diagnosis, an example would be the ELISA test for HIV. So let's do a brief kind of general overview of what exactly is going on and how we produce monoclonal antibodies. So first, we need to initiate an immune response. And to do that, we're going to incorporate an antigen into the body of an organism. And as we already know, when you do that, the body is going to respond. And it's going to respond in producing antibodies. Specifically, we're going to start producing specific B cells because we know that those B cells are then going to become plasma cells to produce antibodies. So when we have these B cells produced because of the antigen that is present, scientists are going to fuse the B cells with a tumor cell. And it's going to do that to help replicate the B cells and therefore make a bunch of antibodies. So when those B cells are combined with a tumor cell, you're going to get a hybridoma. And a hybridoma is going to make a large number of clones and therefore make a large number of antibodies specific to that original antigen. Pretty interesting stuff. So let's take a look more in depthly of how monoclonal antibodies are produced and how they are then used to help humans. So the production of monoclonal antibodies, another quick summary. So again, we're first using the antigen and putting it into, let's say, a mouse. The mouse's immune system is going to produce the B plasma cells, which are going to have antibodies on them. They're going to be fused with a tumor cell, so then we can get them replicating, which will then become, or is called now, a hybridoma. And this hybridoma is going to supply us with a bunch of antibodies against that original antigen. And what's even more fascinating is that scientists have found a way to also use monoclonal antibodies not only to fight infection like pathogens, but also to fight cancer cells, which is incredible. So taking a look here, what is amazing is that scientists were able to now use these monoclonal antibodies and make them specific to antigens on cancer cells. And not only were they specific to antigens of cancer cells, but scientists found a way to also, on the bottom of the antibody, either connect or attach drugs or radioactive material so it brings it directly to a cancer cell to destroy it. Absolutely amazing, crazy, awesome stuff that scientists are able to do with monoclonal antibodies. So here are just some examples of the use of monoclonal antibodies. And again, if you would like to go over the steps one more time and just read through, the steps are here that you can also rate them wherever you have space for that first slide on monoclonal antibodies and the slide that has the mouse on them if you would like to just annotate this picture with the notes that you have here. What I'm going to now ask you to do is go back to this first slide and watch this 4-minute video on monoclonal antibodies. Again, it'll be a nice general overview for you so you get the full understanding of how monoclonal antibodies are made in the lab. So now that we have an idea of monoclonal antibodies, what we're going to be doing next is taking a look at how monoclonal antibodies can help with pregnancy tests, breast cancer treatment, and Ebola treatment. So if you click on this link, it is the same document that you'll find in Google Classroom, and you're going to be watching a couple of videos to see how exactly monoclonal antibodies can be used to help treat breast cancer, how they're used in pregnancy tests, and then you're going to do some research and take a look at how monoclonal antibodies can affect or help treat Ebola. So a very interesting topic today. If you have any questions, please feel free to email me or join the Google Hangout. I hope you're all doing well. I miss you all so much and have a great rest of your day.